Very good morning to all. Today we have to see about the introduction to biostatistics. I am Dr. J. Vijayakumar, Assistant Professor of Biotechnology, INR Janagamal College, Sivagasi. So here I have to share something about introduction to biostatistics. It is one of the very important topic of the biostatistics. That is one of the main subject for BSc Biotechnology student of ANJAC. So before going to study biostatistics, the students must know about the basic need of biostatistics. What is the definition of biostatistics? See, it is defined as the science of collection, monitoring, analysis, and interpretation of facts and numbers connected with biology. See, it is otherwise called biometrics or biological measurement. Say example, in estimation of protein from the sample, we can able to collect the data of the amount of protein in mg per ml concentration through experimentation studies. The data must be entered into the suitable table of the Excel spreadsheet for making of any arrangement systematically before going to adapt any particular method of statistics for interpretation studies. Here lower and in a table you see the lower and higher amount of the protein that is being subjected to interpretation by statistical method. Next you see the concept of biostatistics. Why the people are highly interested in biostatistics? Because of nowadays it is necessary for solving of many research problems. So that's why all the people are considering of biostatistics subject as one of the very important tool for many biological research, including of public health, medicine, environment, so on. So within the data, you are unable to present it in front of others for understanding. The people are unable to understand about the data. What does it mean? So that's why. So there is a need to apply this suitable method of biostatistics to present the data systematically for the people understanding. In case of the population study, so by here you can able to utilize the method for the studying of the group of individual from the population. So population, which means the study of the group of individual from the entire population. Then data. So data is the value which is being collected as a group from the population. So it may be collected from the biological experiment. Then we can able to categorize it as primary and secondary data based on our interest. So if you have collected the data freshly as a investigator, so that is being considered as primary data. The secondary data may be collected from secondary source like magazines, published papers from the reputed journals or from any book sources. Here the sampling from the entire population, that's suitable for taking out of or getting of that value as a data. So here the sample, what does it mean? This sample. Sample is nothing but a small fraction of the population. So once you are getting of sample from the population, it is known to be sampling. So there are many variables from the individual data collected from the experimentation. Say example, size of the fruit. We may get it different sizes of the fruits as a variables. Here each and every variables of the data are not same. Okay, here so you can able to consider the individual items as the variables. So variables are highly applicable for mathematical modeling, statistical modeling, and experimental biological sciences. So come to see about the types of the variables. So there are many types in variables. First one is independent variable. Here each and every values are not dependent from one another. That is nothing but experimental or predator variables. So it is used to determine the causes in order to observe the effect on a dependent variable. So here the independent variables are being utilized based on the observance of the effect from the dependent variable. Without any dependent variable, we cannot study the independent variable. Say example, concentration of macromolecules in rat. So each and every concentration of molecule are differed from one another of the rat or any animal. So that is known to be independent variable. There is no particular relationship with each and every data. What is dependent variable? So one value which depend on other values. So each and every values are highly related with each other. It is otherwise called a response variable. So it is dependent on the independent variable. Say example, effect of nutrients in health condition of rat. So the nutrient level is necessary for the improvement of health of the rat. Here the data are highly dependent one. Come to see about the third method, third variable, qualitative variable. Qualitative, we are unable to measure the variables of the data. So that is nothing but attribute the variable or immeasurable the variable. Say example, color of the flower, banding, intensity of DNA in gel. Color of the flower is not possible for any estimation or quantification. In this case, you can able to observe the visually about the coloring pattern of the flower. Then only you can able to state it well systematically in a order. That's it. But we are unable to quantify it in a proper way. In this case, the intensity of the DNA binding pattern is also differed, but we are unable to estimate it well. The binding pattern may be observed for the measuring of actual sizes of the DNA in each and every line of the gel. Next variable is quantitative. That's very important for further statistical analysis of the data. Here it is consisting of two types. First one is discontinuous variable. Second one is continuous variable. The collected values are the rounded one, or whole numbers are being considered for discontinuous variables. But if the numbers are decimal, that is considered to be continuous variables. Say example 17.5, the point something values are decimal numbers. It is not undergone discontinuous variable. Instead of that, we may categorize it as continuous variables. So in this case, you have to categorize the data whether it is undergone discontinuous or continuous the variables. Then only we can able to adopt the best method of the statistics for analysis and interpretation. Finally, you should know what are the notations followed in statistical the methods. I have listed out some of the symbols based on the parameters of statistics. First one is the mean. So here the x bar is the symbol of mean. X is the symbol of variables. Then sigma is the symbol of standard deviation. The F denotes frequency. H0, it is denoting of null hypothesis. P stands for probability. Sum of simple is designating of the summation. Then the chi-square symbol is also they given for chi-square test. So the other symbols are also available, but basically the biology people should understand about what are the symbols of the variable formula in biostatistics. That's very important. Um, here here I have said something about basic or need of biostatistics, introduction to biostatistics. I think it is very helpful for the students and their understanding. No, if the students are understood about these basic information, then only they may study in depth about each and every statistical analysis for analyzing of their own data. The PG students may collect the data through the experimenting of anything. So the experimented data or collected data through the experimentation, so which is being subjected to the statistical analysis before going to interpret it well for proper presentation in the IOs or the preparation of the dissertation or thesis. If anybody is not analyzing of their data with the suitable method of biostatistics, the people unable to understand about your research work. So student, say biology student, they should know about the statistics method. Then only we may adopt the method for any analysis and interpretation. Here, I think the students are understood about this particular topic. If you have any query or clarification, you can able to ask it through sending of the mail to my mail ID. My mail ID is Dr. J. Vijayakumar 15 at gmail.com. So, thank you, one and all, for listening to my lecture.